Okay. Good morning, everyone, and welcome to another session of the Netpore webinar. So today's speaker is Bruno Miranda. Bruno is a researcher at the Institute of Applied Science and Intelligent System of the National Research Council in Italy, in the city of Naples. And he received his PhD in Information and Communication Technology for Health at the University of Naples, Federico II. And his research interests are devoted to the design, fabrication, and characterization, as well as the functionalization of hybrid metallic and dielectric nanostructured materials for optical biosensing. He gained experience in plasmonic nanoparticles, polymer nanocomposites for silicon, and photonic crystals for biomedical applications. In today's webinar, Bruno will discuss porosilicon biosensors, concerning fabrication, characterization, and functionalization strategies for diagnostic purposes. So please, Bruno, you can share your screen.
Good morning, everyone. Can you hear me?
Yes.
Okay. First thing is okay. Let's try with the second one. Okay. Can you see the slides? Spoiler.
Yes.
Okay. Okay.
Yes, we can see the full screen.
Okay.
Thank you.
So, good morning, everyone, again. Today I'm going to talk about porosilicon biosensors, and thank you, Eleonora, for your nice introduction. So she said almost everything here, just a few notes about my experience. So I graduated at the University of Naples Federico II in Industrial Bioengineering, and I did a research internship at the Okinawa Institute of Science and Technology in Japan. And then I started my PhD, and already everything about this. I work at the Institute of Applied Sciences and Intelligent Systems in Naples, and the main activities of this group is imaging, microscopy and optical analysis, applied photonics and optoelectronics, and especially in my case, we work on functional devices, sensors and biosystems. Today, I'm going to provide you a brief introduction on optical biosensors, especially the ones based on porosilicon. I will show how we fabricate porosilicon and what are the general properties of porosilicon devices. I will show how it is possible to characterize and functionalize porosilicon. I will go to some of the applications we are carrying out in our group, and I will finish with conclusions and perspective. So, first of all, what is a biosensor? The World Health Organization has standardized the classification of biosensors in general with many criteria in the acronym REASSURED. So, a biosensor must be a real-time device, so providing real-time signals and outputs, should guarantee easy sampling, so to meet the compliance of patients. It should be affordable in terms of costs. It should be very sensitive and specific towards the analyte of interest. It should be user-friendly, rapid and robust, and also deliverable, so it means portable and equipment-free. These are the main criteria to obtain a biosensor, but how far are we from these? Generally, nowadays, we know that there are many biosensors, but especially for glucose monitoring, for example, for diabetes, but not all the criteria are always respected. So there is strong efforts from many researchers and many groups to guarantee that the final device you design and sell in the end to the patient meets all these criteria. But what is a biosensor? A biosensor is a device that converts a chemical interaction into an electric signal with data that is displayed or that can be seen by naked eye. What are the typical elements? We have the analyte of interest, that is the first target that you want to detect with high sensitivity and specificity, and you can do it by using a biorecognition element that is specific for the analyte of interest. There are many types of bioelements. They can be enzymes, antibodies, but also entire cells that can act as biorecognition element. Then we need the transduction of the chemical interaction into an electrical signal. This transduction can be electrochemical, piezoelectric, optical, or calorimetric. Then there is all the electronics part that send the signal to a display. Okay? How the idea of making a biosensor was explained during time. So initially in the 90s, in the last century, there were different types of laboratory-based assays. The first one, the most simple, the simplest, was the immunoassay. For example, here you can see a sandwich immunoassay in which you have a primary antibody that directly recognizes the antigen. Then you have again this antibody that recognizes another epitope in the antigen, and then you have a secondary antibody that is generally labeled with a fluorescent dye. But also you have some other strategies later in time, for example, apta sensors, in which the biorecognition element is an aptamer, that is modified that is an element that is synthetic, okay, and is capable of recognizing its target and quench or enhance its fluorescence according to a specific recognition of the target. Also later, the recognition element became polymers that could recognize the target by adopting the shape, the conformational shape of the target. And in this case, you have a mechanical or physical entrapment of the target inside the polymer that in this case, of course, reduces the costs because it does not require any purification or external synthesis. But also later there has been the development of DNA-based, so nucleic acid-based recognition elements that can be also engineered in order to form hairpins and be very selective for their target. But then all these strategies were implemented, integrated into portable sensors. The first example is the lateral flow assay, in which you have that the recognition happens on a paper-based element. So you have very portable and low-cost material on which the antibodies, for example, can be immobilized. And then you have the conjugation pad where, for example, generally there are gold nanoparticles that interact with the target if it is there or not. And then you have test lines and control lines. One example for this, of course, in the last years was the COVID test, and we have seen that it has been very useful for large-scale screening, so on the whole population. Then we can have more sophisticated things involving microfluidics. So, for example, DNA amplification, RNA amplification on microfluidics, but also integrated microfluidic platforms. Subsequently, we are trying to integrate these systems into with smartphones that are available to everyone, and smartphone can become, for example, the light, so the source of light, and also the detector, especially for optical systems. Nowadays, we are moving toward more sophisticated things such as CRISPR-Cas integrated chips for gene detection. But also, we are moving towards wearable sensors. The first example was tooth sensor for bacterial detection, or flexible graphene nanocomposite field effect transistor that could be, because of its flexible nature, because of the presence of polymers, basically, you have wearable sensors that can be adapted to the skin, for example, for thermal monitoring, but also implantable devices for drug release monitoring and implantable intraocular biosensors. So as you can see, there has been a strong effort to improve the technologies behind the simple sandwich immunoassay of 1960s. In particular, in our group, we work with optical biosensors in which the transduction element is an optical device. So light is converted into an electrical signal. And in particular, today, I will show you the advantages of using porous silicon as an optical biosensor. But what is porous silicon? Basically, silicon is a material that can be, with nanofabrication strategies, can be reduced to nanoscale. So some pores at the nanoscale can be obtained with different techniques. In our group, we use electrochemical etching. In particular, the advantages of porous silicon are high surface area, tunable optical properties, chemical tunability, biocompatibility, and also low costs. And what are the main applications? Biosensing, but also bioimaging, because to porous silicon we can give also photoluminescence properties. As I said before, silicon is a semiconductor that is an indirect bandgap semiconductor, but it can be doped to provide to it different electrical and optical properties. It can be N-type doped, and so it means that there will be free to move electrons with a negative charge, or p-type doping, in which we will remove some electrons by substituting other silicon atoms with other atoms, such as boron or gallium. And in this case, we say that it has overall a positive charge. Of course, it's not a positive charge, but it's lack of electrons. So the first to discover porous silicon was Harris in 1956. Then Canham in 1990s discovered the photoluminescence of porous silicon, and Richter in 1991 discovered the electroluminescence of porous silicon with solid state contact. How do we do the fabrication of porous silicon with electrochemical etching? So we have this chamber in which there is a platinum electrode and a cathode on the base, and we have the flow of a current with the programmable current control in which we have that this electrolyte that is generally made with hydrofluoric acid, but we will see it depends on the doping of the material, but also on the properties, the final properties that you want to have. But according to this, you can have the flow of the current to this electrolyte that goes on the surface of silicon wafer, and according to the doping, you will have the formation of pores according to this chemical reaction. The final result is a porous silicon substrate that, as you can see here, can have different optical properties according to its pore size, dimension, and doping. How is the cell made? So these are two different type of cells that we have in our lab. We have the cell in which we put the silicon wafer, in this case, so entirely, or a piece of the silicon wafer. We have the platinum cathode and the anode, and we close this chamber, and according to the current density, the concentration of electrolyte, the of hydrofluoric acid, of the electrolyte composition can be made with ethanol or other solvents, but also on the doping, we can have different curves. This curve is generally made to characterize your silicon wafer. So you will have an increase in current according to the applied voltage. And you can obtain this IV graph in which you have the first region in which you have the pore formation. Then if you increase the current and the potential, we have a transition region in which there is no pore formation, but also there is no electropolishing. And finally, there is the third region in which there is electropolishing. What is electropolishing? Basically, you you come back to the flat silicon. Generally, electropolishing is used both to clean the surface before the pore formation or to detach the obtained membrane. UPAC classification has been done for the classification of porous silicon. Basically, you have macroporous silicon if their pore size is higher than 50 nanometers, mesoporous silicon in between 2 nanometers and 50 nanometers, and microporous silicon if it is lower than 2 nanometers. Generally, we use low-doped N-type silicon for non-photoluminescent macroporous silicon, while we use low-doped P-type silicon for photoluminescent mesoporous or microporous silicon. How you describe the optical properties of porous silicon? Basically, you have that according to the increasing in porosity, to have an increase in porosity, you must apply a higher current. Okay, so with the increase in porosity, what you expect is that you have a decrease in the refractive index of the material. Basically, you can describe the porous silicon monolayer as an double interface. So you have here the medium that can be air, but also water, solvents, according to where you put your porous silicon device. Then you have an average between between silicon and the medium, and here you have the substrate that is simply silicon. So how can you model the refractive index? You can use the Bruggerman model, in which there is an approximation in which you have the refractive index of silicon, the refractive index of the medium that are well known, and knowing the porosity that you can derive from this curve, you can extract the average, the approximate refractive index in the membrane. And why it is important to know the refractive index of porous silicon is because you can tune the optical properties according to the different geometries that you want to create. For example, if you have a simple monolayer with a defined porosity, you can predict what its reflectance will be. And for example, with a monolayer, you have interference and constructing but destructive interferences. So you have this increase and decrease of the reflectance according to the wavelength, as a function of the wavelength. Or you can have a Bragg mirror here when you have one layer of porous silicon with high porosity, one layer of porous silicon with low porosity, then again one layer with high porosity and another one with low porosity. So you alternate different layers of porous silicon with different porosity, and you can obtain a Bragg mirror in which that acts as a bandpass filter. Or you can have an optical microcavity with very sharp resonances, as the case C. I don't know if you can see the pointer, but
Yes.
You can see it, okay. You have the optical microgravity here, in which you have very sharp dip in the reflectance spectrum that can be very sensitive to refractive index variations for biosensing, or you can have very complex shapes such as this one, in which you can tune the optical properties according to what you need for your biosensor. How can you do it? Basically, for example, to make a double layer in which you have the convolution of two reflectance of these two layers, basically, you apply a high current and you obtain the high porosity layer, and then you apply a lower current to have this, the other one on the bottom. And the final results, of course, will affect the optical properties. It is important to have the programmable current control, in which you can tune the current you apply to tune the final properties of your device. In our case, I will show that we basically work with porous silicon monolayer, that is also called Fabry-Perot interferometer, that can be described by this formula, in which lambda L represents the maxima wavelength position, M is the spectral order of the fringe, L is the thickness of the film. So I can characterize the thickness of this film by using, for example, AFM or scanning electron microscopy by tilting the sample and measuring the height of the pores, and knowing with the Brueggemann model the porous silicon refractive index. So we can predict this shape and tune where the maxima positions of this spectra will be. What is the advantage of C-monolayers in biosensing is that we have a simple and fast fabrication strategy. The molecules for biosensing purposes are fastly diffusing in a monolayer rather than in 20 layers of porous silicon. So this makes the time fast, so for the interactions and for the calibration curves construction, and the possibility also to detach the membrane for the creation of nanoparticles with photoluminescence properties. Moreover, controlling one layer of porous silicon is much more reproducible than controlling 20 layers. Then, of course, it depends on the final results that you want to get. As I said before, Karam was the first to discover also the photoluminescence of porous silicon, and basically, according to his measurements, the origin of visible photoluminescence is basically due to quantum size effects. So the lower is the dimension of the features of the crystals that creates in the pore, the higher will be the photoluminescence. So you can tune the dimension of these crystals by basically changing the way in which you perform the electrochemical etching. And he was able to obtain very strong photoluminescence under argon atmosphere from red to blue in all the conditions. Now, here, for example, you can see the photoluminescence of porous silicon under UV excitation with an initial porosity of 68%, but this is when you detach from the substrate, so the detached membrane. But what happens if you oxidize this porous silicon? It directly becomes blue. So the photoluminescence is something that is very unstable. So we need several strategies to passivate these structures and try to improve their chemical stability and also their photoluminescence. the two types of structures that we create are these ones. So basically, this is a porous silicon monolayer that is macroporous silicon that we obtain with these conditions, and this is the typical reflectance spectrum that we can both predict and measure with optical setups. And this is the microporous silicon that is instead photoluminescent. Here you can detach this membrane, sonicate, so mechanically break this membrane into nanoparticles with controlled size. And this is the typical photoluminescence spectrum that we can measure with spectrophlorimetry, or also typical setups that are on the optical bench. So you need a UV laser, and you have to shine light at 90 degrees and collect what is the signal of the photoluminescence. How we characterize porous silicon? So as I said, we need a biorecognition element. So to make this biorecognition element available in the pores of porous silicon, we need a functionalization strategy that we can monitor optically with this typical reflectance setup, in which you have a light source, then you have a Y-shaped optical fiber. The light comes on the sample and is reflected back and recollected into the spectrometer. And then you have the computer for the analysis of the spectra. So this is the typical reflectance spectrum. And what happens after functionalization is that you have a shift in the reflectance spectrum, and also you can compute fast Fourier transform to be sure that what you see here is coherent with all the fringes of this reflectance spectrum. So you perform a fast Fourier analysis, and you will have a single peak in which that is much shifted compared to the reflectance spectrum, and basically here you are making a more accurate measure on what is happening overall on your device. But also we can make photoluminescence analysis. So you have a UV laser, then it is photoluminescent. Generally, when we functionalize photoluminescent porous silicon, we have a decrease in the photoluminescence that is due to the quenching effect due to the presence of molecules. But as I said before, porous silicon is very unstable. So the freshly etched porous silicon undergoes chemical instability that causes the degradation of the material because of its oxidation. So the optical properties, for example, generally, if you leave the porous silicon as it is, undergoes a blue shift instead of a red shift because you are losing material. So to avoid this, we need a passivation process that avoids the oxidation or stabilize the oxidation process. And for example, these are the main three strategies that are generally used for the passivation. The first one is thermal oxidation. So here, instead of avoiding oxidation, you totally oxidize the surface of the pores. In this way, you have silicon dioxide on the top layer of the pores, and there is no chance anymore to degrade this material because silicon dioxide is very stable, and you do this with a thermal treatment. Or you can have some other strategies such as hydrosilylation and hydrocarbonization, in which you substitute the very unstable CH groups with the CC SiC, silicon carbide, so silicon carbon bonds that are very much more stable and also offer the possibility to link some other molecules on the top. So this is a typical functionalization strategy. So for example, the thermal oxidized porous silicon can be silanized with aminopropyl thietoxysilane, that is APTES or APTMES, that provides an amino group in the end that is very useful for the linking, for the binding of proteins. Or you can have, after hydrosilylation, the pegylation of porous silicon with carbonimide chemistry. That is a very common click chemistry in which you have the peptide bond between a carboxylic group and a mini group. And in the final here, you will have generally again an aminic group at the end. Or after one of these two strategies, you can go and bind, for example, an antibody. Now, you can bind directly on APTES or on PEG, the antibody, because it is it has a lot of carboxylic group to interact with the surface, but in this case, it will be not oriented. As you know, or you may know, antibody has two regions. The Fab region, that is the arm, and it has two arms, so it has two Fab regions, and there is an FC region that is constant, and this part is the one that is specific for the antigen. Now, if the antibody is not oriented, it may be not capable of interacting with its antigen. So generally what we do is adding a protein A layer that is recognized by the constant portion of the antibody, and in this way you have a perfect orientation of the antibody that is free to bind its antigen. This is very important because in this way we are sure that each antibody is capable of interacting with two antigens. But there are also some other passivation strategies and also functionalization strategies that involves polymers that can be infiltrated, coated, can form a capping on the surface of the pores, or can embed the entire freestanding membrane. And also, what is the advantage of using polymers is their cost effectiveness, high processability, and also the possibility to adapt to non-planar substrates. Moreover, for example, if you have a flexible device, a polymeric device, you can guarantee the direct in situ and in vivo collection of samples, and this integration in more complex systems. So we are also working in this direction because we know how to deal with also polymers. I will show now some of the applications of our group. The first one is the design, fabrication, and characterization of an hybrid graphene oxide porous silicon biosensor for Brugada syndrome detection that can cause. This syndrome is very difficult to handle because it can cause heart attacks. For example, it's very famous the story of this soccer player that was, while playing his soccer football match, he was dying because of a heart attack. But basically he had this syndrome that is due to point mutation in this gene. So you have just one mutation in the DNA of this gene, and in our case, we planned to use peptide nucleic acid as a biorecognition element to monitor the DNA and verify the one base mismatch between this gene in order to recognize this mutation. And what is the advantage of using peptide nucleic acid instead of DNA? is that it has more strong and specific hybridization to the single strand of DNA, but also its charge is neutral. And this is very important because in this way there is no charge effect on the surface of porous silicon. And the only charge that we will have is with its complementary DNA. How we planned to do this? We wanted to have a double check. So we wanted to have both photoluminescence and reflectance to monitor the interaction of the PNA on our sensor to immobilize the biorecognition element. And so we decided to study graphene oxide photoluminescence properties and use the macroporous silicon that is not photoluminescent. So we grafted covalently the graphene oxide on porous silicon, and we obtain a multiparametric hybrid device. The surface modification was the one that I showed before. So you have the SH, the porous silicon, you hydrosilylate, pegylate, and then you functionalize covalently with carbonimide chemistry the graphene oxide on the top of this surface, and you immobilize PNA on the top of graphene oxide. Finally, you make to interact the PNA with the DNA, and you obtain your final device. So the hydrosilylation process was performed to have the final carboxylic group at the end, to which graphene oxide was attached. So we monitored each step, and here you have the graphene oxide infiltration. Before and after graphene oxide, you have a red shift here in the reflectance spectrum, but also what is important is that the macroporous silicon from non-photoluminescence becomes photoluminescent with a modulation. This modulation is due to the interference with the reflectance spectrum. So this means that graphene oxide was directly inside the pore. Otherwise, its photoluminescence would have been smooth. Instead, you have these fringes due to the presence of the porous silicon. We checked that also to verify the functionalization with graphene oxide, we also used labeled protein A to check with the confocal microscopy that the protein A was inside the pores. And as you can see, since the thickness of our membrane is 2 microns, we have a maximum around 1 micron. So this is important to verify that as the protein A, also the graphene oxide was inside the pores. Then here we have the PNA-DNA hybridization. So after GO, we have PNA, and we have a further red shift in the reflectance spectrum. And what it's important to see here is the quenching of the photoluminescence due of graphene oxide due to the presence of PNA. Finally, we have DNA hybridization with different concentrations. Okay, and we constructed our calibration curve. What is important is that the mismatched DNA with one base mutation did not cause any shift. So it means that PNA was strongly selective for its target. These are some SEM images to show which the optical, the porosity of our porous silicon, and also here we have some controls that we performed to verify that the non-complementary DNA was not able to verify under fluorescence microscopy. So we labeled the non-complementary and complementary DNA with the same fluorescent dye, and as you can see here, in presence of the complementary DNA with the PNA, we have a strong fluorescence, also working as a function of the concentration. Another recent application that our PhD student Valeria Nocerino is carrying out is the monitoring of interleukin 6, that is a cytokine that plays a central role in the inflammatory response of tissues and also in the initiation and progression of atherosclerotic process, is to use this molecule by detecting its presence in artificial serum with molecular imprinted polymers. Basically, here what you have is a polymer that specifically is made to interact with interleukin 6. Then we have template removal, and the final result is a hold polymer in which that has the typical shape of interleukin 6, for example. And what we can see here, these are very promising results. We can see that with the molecular imprinted polymer, we have a red shift, okay, of the reflectance spectrum. Then we have the washing step, and we have a blue shift because we are removing the template from the molecule, from the molecular imprinting polymer, and then we have a different increasing concentration of interleukin 6 that cause a further red shift. So what we have to monitor is from the washing step to the interleukin 6. The maximum shift ideally that we could see is the initial one, in which all the molecular imprinted polymers was imprinted with the template protein. And as we can see here, we have this preliminary data, and we are achieving very promising results. I don't know how much time, Eleonora, do I still have?
I think that if you can finish in 5 minutes, that would be nice. Thank you.
Okay, thank you. And okay, very fast. As I told before, we have photoluminescent nanoparticles that are obtained from photoluminescent porous silicon. We mechanically detach the membrane and ultrasonicate it in order to break this membrane into nanoparticles. What is the advantage of using porous silicon nanoparticles as labels in bioimaging is that they are non-toxic. Instead, the commonly employed semiconductors such as cadmium selenide and cadmium tellurium are very toxic for in vivo applications. Instead, porous silicon is highly biocompatible and biodegradable. But what is its main limitation is its instability. So we developed functionalization strategies in order to passivate these nanoparticles and stabilize its photoluminescence. This is the typical suspension that we obtain that is very photoluminescent under UV light. And very fast, we performed some stability test. We performed hydrosilylation on these porous silicon nanoparticles. So while the porous silicon nanoparticles exhibited an increase in their size, meaning that they were aggregating in the medium, and also its photoluminescence was quenching during time, hydrosilulated porous silicon nanoparticles remained very stable in size and very stable also in PBS in terms of their photoluminescence. Now, we decided to test these nanoparticles on Hydra vulgaris, that is a very small animal that was done in collaboration with the group of Claudia Tortiglione, and its safety, its health can be monitored with this Wilbis classification. So 10 means that it is very well, it's staying very well with the exposure to these nanoparticles. 0 means that it is going under that. What we did was testing the hydroxylated nanoparticles as they were. Of course, the animal was in a good condition, but the problem is that the nanoparticles were not internalizing because of their negative charge. So we decided to modify them with polylysine that is positive, and in this case, as you can see here initially, we can see that the nanoparticles are inside. To be sure that they were inside, we also performed some in vivo time-gated imaging in which you can see the autofluorescence of tissues that has a very lower, a much lower decay length, decay time, so in the order of nanoseconds, while the nanoparticles have microseconds in decay, in the decay of the photoluminescence in the time. And as you can see here, the photoluminescence of the nanoparticles remains inside. Also, what we can see from these images is the overlap between the tentacles of the animal and the photoluminescence of the nanoparticles. Okay, I jump to the conclusion. What we have shown is the potential of porous silicon biosensors for various biomedical applications for their properties, including large surface area, tunable pore size, and biocompatibility. Porous silicon biosensors can detect various biological analytes, such as proteins, nucleic acids, and so on, and they could be the future for point-of-care diagnostic, environmental monitoring, and personalized medicine. However, what we expect for the future is the integration of these devices into more complex systems, including light-emitting diodes and photodetectors, and a bigger collaboration with all the people from the science, from clinicians to doctors to biotechnology to also industrial partners, because it's important that since this is a very multidisciplinary field, everyone contributes to the development of these platforms. I want to thank all the people working on these activities, starting from the nanobiosystem groups guided by Dr. Luca De Stefano, and all the people that are working on the experiments that and have been working during the years on these experiments, especially Valeria Nocerino, Rosalba Moretta, and Chiara Schiattarella. And I want to thank you for your kind attention. Sorry if I was very long, and the organizers for giving me the opportunity to present this research line.
Thank you very much, Bruno, for this comprehensive overview of sensors and of biosensors and porous silicon application, and for the interesting application that you presented. There is time for question, so if anyone has a question, please raise your hand or speak directly. Okay, Frederic, please.
Yes, thank you very much, Eleonora. Thank you, Bruno, for your very nice overview of the topic and of your work. It was really interesting. I have a couple of questions. I'm just curious, in your sensor system, the biosensors one, you have modified with PEG. You have a PEGylation step in your surface modification. And I was wondering, what is the role of the PEG in this case?
It's a linker between, if I'm not wrong, between, I can show the slide again, just a sec. But it is a linker between the passivation and the graphene oxide.
Okay. And don't you have any problem because it's quite big PEG? Do you have to optimize?
No, we have the 600.
Okay.
It's a small PEG. It's a very short PEG. It's 600 Dalton.
Okay. Thank you.
Thank you.
Can I ask another question?
Yes, of course.
I'm just curious. In the other sensor system, you use the template approach. I was just wondering, how do you remove your template from the polymer?
Okay. It is since it is not a covalent binding, but it's just a conformation. Okay, a structural. Okay, interaction. You can simply use an an AOH, so a very basic buffer. So I don't know, the sodium hydroxide. Yes, sodium hydroxide is the one that we were using to detach at, I don't know, it was 5 millimolar, something like this concentration. And in this way, you see the detach of the template from the polymer that instead remains in the porous silicon.
Okay, thank you.
You're
Thank
welcome.
you a lot. And say hi to Luca for me.
Okay.
Thank you.
I will do it.
Thank you very much.
Thank you.
Thank you, Bruno. There is another question from Jonas.
Hello. Thank you, Bruno, for nice presentation.
Thank you.
Actually, I'm not strong in biosensing. I'm more interested in preparation of silicon. What about complicated word, reproducibility? I mean, from sample to sample, can you get the same feature, the same luminescence, the same porosity? I mean,
okay, on average, yes, it depends. So we always check the conditions, so the buffer condition, the electrolyte solution, the way in which we treat the porous silicon. So if you pay attention to all these things, we have a good reproducibility. We can say 10% variability on the signals. Okay.
Fine. That's fine. May I another question?
And generally, we discard the ones that we don't like, of course.
Yeah, so that's why it's hard to work with porous silicon.
Yeah, of course, it's a
bottom. It has its own character.
It's a large scale approach, so it's a very low cost. So it has some variability, of course, but we can handle it by making a lot of statistics on that.
That's right. May I another question?
Yes, thank you.
Sure.
Is it possible to create not straight columns of silicon, but cone-like columns by changing current during time? But your graph was illustrating that the current changes in time already.
Yeah. Okay.
I mean, porosity, change of porosity with thickness.
Okay. The problem in the electrochemical etching is not the right strategy, I think, to tune a lot the way in which the pore is formed, but there are some other strategies that you can use for making cone-shaped structure that is not electrochemical etching, but MACE, so metal-assisted chemical etching, in which you can tune the way in which the pillar is formed by changing the dimension of the particles that you are putting on the top. But with electrochemical etching, I am afraid that this is a kind of unfeasible, because what masks the pore formation is the dopant. Okay, so from there I need to show the slide. Just a sec, sorry. Okay. This one. Okay, so here you have, this is the doped silicon, and then the pore is formed starting from these points. Okay, so I think it's what you can do is not keeping constant we should try, by the way. This is a good idea. But I am not sure because I have never tried. But basically here, instead of keeping constant this, we could decrease it
in time,
in time, and maybe in this case it could work. But I have never tried, so maybe I will let you know if there will be time next time.
It would be very interesting.
Yeah,
if we get some new results.
Yeah.
Thank you very much.
Thank you. Thank you.
Nice.
Thank you, Bruno. There is one last question from Olena, who wrote, what about long-term use and stability of your porosilicon substrates, I believe?
if you don't passivate the surface of porous silicon, of course, maybe in 1 week it could degrade also in air condition. But if you if you want to have the freshly prepared porous silicon, you can store it also for long times under vacuum or under argon, okay, or nitrogen gases. It should be quite stable. But if you want to use it in normal condition, ambient condition, you have to passivate with one of the passivation strategies I showed before. And in this case, we do not see big variation in the optical response of the device.
Okay.
I hope I answered the question because...
Thank you very much, Bruno, and thank you again for your very nice talk, and for also for waiting 1 month to give your talk. And I say goodbye to everyone. So the next webinar will be in 1 month on June 13, I believe, and so you can check on the Netpore website for this. Okay, thank you. Have a good day.
Thank you. Bye bye.
Bye bye.
Thank you very much.